The title of their joint seminar today is Scooped! Estimating Rewards for Priority in Science. Please join me in giving Carolyn and Ryan a warm IQB welcome. Ryan, Carolyn, the floor is yours.
Thank you very much. Sorry, I'm sorting out my Zoom here. Okay, can you see my screen? I'm having a bit of a...
We see everything perfectly. You were much better a moment ago when you were big.
Well, I said I seem to have lost my, just a second, let me sort this out.
Too many windows.
Yeah, that's a problem.
Presentation.
You're good now. You're good now. Yep. Why don't you move your cursor just to make sure we can see it? Yep, we can see your cursor.
Okay.
Thank you. Go
All
for
right.
it.
Great. So I'm very excited to be here, and we are, and this is an unusual seminar for us. We've presented this mostly to economists and social scientists. So this is the first time we've sort of bridged the gap and come over to present for life scientists and people interested in quantitative biology. And so we're very grateful to get your feedback and questions at the end, and let us know if anything doesn't seem to resonate with you, or any areas where you think we can improve. So I'll start with as a somewhat broad discussion. I've added this slide here so you understand why we're coming at this as economists. The economics of innovation is a really interesting area to study, in part because new knowledge is an important good for us to produce, but it's sort of different than a usual market good that we usually study in economics, like cars or Apple computers or something like that. So a scientific finding, we say, is a public good, and which can cause market failures. The reason we call it that is there's two criteria. One is that a new scientific finding is non-rival, meaning it does not diminish in value after use. Many different people can read a scientific paper and get value from it, unlike an ice cream cone where only one or maybe two people can value it. It's also non-excludable, meaning it's difficult to control the use of that idea. So anyone can use it without paying for it because it's so cheap to share new ideas. If I sort of read a scientific article, I can tell my friends what I learned, and they can then benefit from that use, and they didn't have to pay for it. And so that's makes it challenging then to encourage people to produce innovations. We say that public goods are underprovided in the market. So for example, profit-seeking firms don't participate in a lot of basic science research because they're able to free ride off of other people who do it. So another example is like the protein data bank itself is a public good. They, of course, have a lot of valuable insights and sort of provide a lot of new data to the world that's useful. But private firms don't seem to participate much in providing it because it's hard to sell that knowledge to the world. So how do we then, encourage innovation? If this is really valuable for the world, what systems do we have to incentivize people to figure out new ideas and make scientific discoveries? One thing we do is patents, which encourages development of applied innovations. But of course, that only works for products that are ready for commercialization. Science is typically sort of earlier in the process. Basic science research has high spillover values, but it's not necessarily ready to go out and sell. And so we say that basic research has been mostly funded by governments in an academic setting. In our talk today, we're going to be thinking a little bit more narrowly on what incentivizes scientists to participate and how do we sort of reward them for the knowledge that they come up with. So this is actually a sociologist of science who was writing about this quite a long time ago. He says that, in short, property rights in science become whittled down to just this one: the recognition by others of the scientist's distinctive part in having brought the result into being. That's Robert K. Merton in 1957. And so what he's talking about really is the idea that scientists are trying to build a reputation. And that's how often they get rewarded for their ideas. When they publish new findings, people recognize that they're sort of a credible source of new scientific knowledge, and they can turn that reputation into grant funding and salaries and so forth. So we care a lot about what the community thinks about our research. In particular, we'll define priority as the credit given to the individual who first makes a scientific discovery. And I emphasize the word first because we often see cases where two teams are competing to be the first to publish some new idea. They might be working kind of in tandem or in parallel, excuse me, and there's a sense that whoever publishes first is going to get the outsized share of the credit. So we not only sort of care of what you've produced, but also we care about rewarding someone who's done it first. And you can think about historical cases of priority disputes in science. So first, Newton and Leibniz, all the way back in the invention of calculus, had a very heated debate over who should get credit for that discovery. Darwin and Wallace, similarly with natural selection, and all the way up until recently, this is an example of the proof of the Poincaré conjecture had sort of a protracted dispute over who should get credit for doing it first. But it's not just the famous scientists who worry about priority. In our day-to-day life, as researchers, we worry about being the first to publish. There's a concern that if you've spent months or even years working on a project, if someone scoops you, as you say, or kind of preempts your publication, you may get much less credit than you would have had your publication been first. We think that might drive a lot of how scientists behave. It encourages them to work quickly and also to publish their findings to the world. It also might affect how they, strategically choose projects, or it affects the choices they make as a researcher. But we don't have a lot of empirical evidence about how these races play out. In particular, in our study, we want to ask, what's the causal effect of getting scooped? In other words, if we were to just sort of isolate these races and find two teams who are on the brink of publishing, how much extra credit do you get for being first? In terms of the short run effect on the project, are you more likely to publish altogether? What's the effect on journal placement and citations? We'll also think about the long run effect on careers, so the future productivity of the scientist and their ability to continue working as a researcher. And then we'll also, towards the end, we'll talk about how the priority reward system might reinforce inequality in science. And so, another way to ask that is the scoop effect equal for high and low reputation teams. This is actually going to be a very nice case where we have these two teams who've done basically the same science, but one might be from Harvard and one may be from some lower ranked university, and we can compare how much credit they get within those races. So to preview what we found in the priority paper gets 54% of the total citations, and the scoop paper gets 46% of the citations. And we'll show you how we use the protein data bank to make these estimates. First, we'll say that that share of that distribution of rewards is much more even than we originally expected. We often think of these kind of as winner take all races, where if you scooped, you kind of get nothing in terms of citations. But in fact, there is quite a bit of credit sharing. The scientists, we actually did a survey of scientists, and they were also more pessimistic. They thought the scoop paper would only get about 26% of the total citations, and so that's much lower than what we actually find. But there is a penalty. I mean, scoop projects are less likely to be published altogether. They're less likely to appear in a top journal. In the next 5 years, though, the scoop scientists, they have about the same number of publications, so we don't see a big discouragement effect from getting scooped, but they do have fewer citations to their subsequent work. So there does seem to be some persistence of the effect of getting scooped on your future the future attention toward your work. Indeed, the priority system does seem to reinforce inequality. The citation penalty is larger for low-ranked teams than it is for high-ranked teams, and we'll show you some results on that. So in terms of our agenda, I usually spend quite a bit more time on the background. When we're talking to economists, we need to teach them basically what structural biology is. Most of you, I think, already know those details, so I'll go through that quickly. I'm going to be very careful to define exactly how we find races in our data and what it means to be scooped. We're going to talk about sort of a very specific definition, and we'll make sure that that's clear. And then we'll show you how we estimate the impact and show the results. The last part here, where it said race to the bottom, competition quality is actually a second paper that Carolyn and I have just released a working paper of. There we're thinking a little bit more broadly about how racing affects the incentive system in science, how it affects the quality of the science that is produced. And so I'm actually going to turn the time over to Carolyn at that point in the last 10 minutes or so to show some of our preliminary results from that research as well. Okay, so let me talk first about a few key empirical challenges. If you wanted to go out and study this sort of phenomenon of scoops and races, what do you need to look for in the data in order to be able to do that? So first, we wanted a setting where there's well-defined problems and, quote, one right answer. So economics, for example, or other social sciences are much harder to do this because our projects are a little more squishy. There's lots of different types of data or models or angles that people are coming at in their papers, and it's less of a clear-cut discovery. Structural biology is going to be perfect for us because everyone has a known objective of discovering a structure or determining its structural coordinates. And so there's kind of a more well-defined finish line for these races. Now, it's also helpful to have an objective measure of scientific proximity. We're going to use the BLAST algorithm and sequence similarity to find cases where two teams were working on the same or very similar proteins. And then we also, I think most importantly, it's very helpful to have a view of potential abandonments prior to publication. So if you think about, one option to study these races would just be to go to the publication data, maybe like the Web of Science or Google Scholar, and just find papers that look very similar. You could do text analysis or something. The problem is that once the first paper is published in a race, the second team can then decide what to do with their research. They could either just push it out, publish it as is. They could change it so it doesn't look much like the original project, or they could abandon it altogether. And they're going to make that choice based on high quality they think their research is, and so forth. So the problem is that then when you compare those two published articles, you may not be getting the full sample of races because people will have abandoned. So we wanted to find a setting where we could view some of those potential abandonments, and we're going to do that with the protein data bank. We won't be able to capture, obviously, all abandonments because often people, cut their projects in the middle of their lab work, but we will be able to see a lot of it right at the end stage of the race. Okay. So we use such structural biology here. So this is my short summary for the economists. As you know, they're trying to determine the molecular structure of proteins, et cetera. And the form is, of course, very important to understand how things in our body work. Certain diseases are important to understand structures, and drug design also requires knowledge about protein structure. I will also say, our data ends in about 2017. And so most of what we're looking at is X-ray crystallography. Of course, there are many newer methods that are becoming popular, but we're mostly, I think about 85 or 90% of our data is for crystallography. Okay, so just to review the key empirical challenges, the structure discoveries are clear answers to concrete questions. That's good for us. The BLAST algorithm we're going to use to quantitatively link projects that are identical. And again, that's a very unique setting here that we couldn't do in most areas of science. And importantly, the PDB, because they require projects to be deposited prior to publication, that allows us to observe some abandonment in the last few months of the project's life. And I would just say, it was quite a revelation for us to come across the protein data bank. We had, been casting about for different areas of science to study this in, and my father-in-law, actually, who's a biochemist, pointed me to this data set, and almost immediately we realized it was going to be very useful for the types of data work we wanted to do. And so, kudos to the PDB for having such organized and useful data, even for economists. So the protein data bank, one big benefit, of course, is that it's very large. So 100,000, or more proteins and very detailed metadata about their projects is pretty unique. It's hard to find an area of science where there's such an organized accounting of projects, and that's going to be useful for our statistical work. Of course, the major scientific journals require that they're submitting that the structures are deposited, so we have a very full accounting. And also the structures are deposited confidentially until article publication. And I'll talk in a minute of how we use that fact in our data. So this, of course, is an example. I picked Jennifer Dowden as one of her Cas9 deposits, as it's been in the news recently. What was exciting for us, again, are the key dates, is that we can see when projects were deposited and then released. Usually, you only see when a paper is published, and it's very hard to know what was happening before that. So we're going to use the deposit date, and we can even use the collection date in the PDB a little bit. And so that's very useful. And of course, then it's also linked to the literature. This is where we get our outcome data. So using these PubMed identifiers, we can know what journal it was published in and how many citations the project received. Okay, so here's sort of a rough model that we show people of the timeline of these projects. Of course, this is happening in the lab, crystallization and structure determination and so forth. This is kind of the area that we're going to be focusing on. So of course, they're writing and submitting a paper, it's under review, and then it's published. The deposit date is, it varies by project, but it usually tends to line up around the time that they're submitting their paper or shortly thereafter. So the team uploads the project, and then it's released a few weeks or months later. Again, this it's sort of confidential here, and that's useful for us. We think that these races are going on in parallel, but you often don't know where another lab is in their progress. It's hard to know whether you're kind of in the front or in behind in these races, because it's a pretty secretive field, and especially the deposit is pretty unknown to your competitors. Now, importantly, as we know, projects are released to the public even if they're not published. So after a year, they're automatically released, and that's what allows us to see abandonments. About 30% of PDB projects or deposits are not linked to a publication, and so we're going to count those as abandoned, and that's again unique. Usually we don't see those projects in other fields of science because they just disappear and never enter into the record. And of course, this time period is usually about 6 months on average. For the very sort of competitive projects, it will be shorter. Now, this is sort of the key slide here to understand what we're doing, how we take all of this data and turn it into something that we can study as a scientific race. So the rules for defining a scoop is first to take two projects with at least 50% blast similarity in sequence similarity and written by different authors. Of course, we're looking for competitive cases. And so if two different teams have a protein that are at least 50% similar, we're going to call them in a race. Now we can change that cutoff. We sort of tried to, we pulled 50% from a few articles of structural biology about similarity and homology and things like that. But we can use a more narrow definition like 90% or 100% to define these races as well. And the results are basically the same. So we take those races, we're going to assert that both projects had been deposited before either project is released. So what we're looking for is a case where the two teams had independently completed the project and had deposited it. And then we can observe which one was released first. And we use the order of the release to define who is the winner of the race, in this case, project A, and who is the scooped team in the race. In this case, that's team B. Okay, and so that's how we define getting scooped, and that's how we're going to compare outcomes. I'll show, one reason that we've made these rules is that we're trying to avoid cases like this, where the first project is released on this date, and then we see another team working on the same project come in, the same protein, come and deposit after that date. So the problem here is that this part then is sort of a selection bias problem. This team has decided to deposit after this release, but we don't know if there were other teams that may have abandoned. So at this point, if they had both deposited, we know that we're picking up any abandonment caused by being scooped. We don't know that here. And so we're going to exclude these from our sample. So we're not considering cases like this. We're just focusing on cases where two teams were sort of at risk of winning the race, and we can observe which one came out first. Okay, so to summarize, we take all of the deposits between 99 and 2017, and then we narrow it down using these three criteria. As I mentioned, that leaves us with about 3,300 deposits in 1,600 races. That's about 5% of the PDB, so 1 out of 20 times your project might be in one of these late stage races, and then about half of that time you would be the scooped team. And so not too common, but enough to be, I think, salient to the scientists involved in this endeavor. So here's an example that, again, we're not trained as biologists, so you'll pardon my inexperience with some of these specifics, but this is the PDX P450 CAM complex, which was published around 2013. This team from UC Irvine deposited in March of 2013, and this competitor came in just a few days later, actually. But this one came out first in June. Their paper came out in Science. Of course, that's a very high impact journal, and they got 52 citations. The Scoop team still ended up in a very good journal, the Journal of Molecular Biology, but of course a much lower impact factor, and they end up having fewer citations. In fact, this is an interesting case where if you go and read this second paper in JMB, they actually cite the first paper and basically admit that they had been scooped and sort of make a note that the structures that they found were very similar. And so that was a very clear case. So our data is a bunch of cases that look something like this. And that's what we're going to do. We're going to compare these to get our citation effects and so forth. Okay, so one important thing we think about as economists is whether we're sort of isolating a causal effect, or we could call this a causal inference challenge, and that is selection bias. One concern would be that if the highly cited labs are also the ones that are so systematically better at winning these races, then if we compare their outcomes and we see that the winning papers always have more citations, that could just be because of selection bias. It could be because those winning teams just get more citations on average. And we're not isolating the true effect of getting scooped. So, ideally you would want almost the winners to be randomly assigned, like a randomized drug trial. That of course is not what happens in the real world, but in our data, we think that the race winners are highly unpredictable. At this late stage in the race, of course, you've heard many stories about projects getting delayed for pretty random reasons, and so it looks like they're highly unpredictable. To show some evidence of this, we use some of the team level variables like their affiliation and their past productivity to predict their citations. We use a lasso regression. And then we want to check that the winners and losers look balanced in terms of their predicted citations. So here, for example, we've taken all of these features and we've predicted how many citations you get on average for the team. And then we plotted them separately for winners and losers. So here, each dot is a matchup in our data. And this is the predicted citations of the winners and of the losers. So if all of these dots were up in this quadrant here, that would mean that the winners are very positively selected. They would be the better teams. But what we see is that it's pretty evenly distributed around this 45-degree axis. In fact, we see a lot of cases where very kind of low-ranked teams are scooping teams from, say, Harvard. And so that means that at this stage, once the projects are completed and we're just sort of seeing who publishes first, that tends to be pretty random. And so we're going to rely on that to do our inference. Okay, so let me go straight then to how we do our estimation. We use pretty straightforward linear regressions, which we tend to do in econometrics. And so our outcomes, like citations, is just regressed on an indicator variable for whether you're scooped. And we can include a lot of control variables here to try to isolate any effects of the differences in labs and so forth, and productivity. But beside, the way to interpret these really is just that we're comparing the means of the winner and loser. We're really just taking the mean citations of the winners and losers and taking the difference. We can include a regression adjustment, but that's sort of the main approach. Okay, so here is our top line results. So if you get scooped, you're 2.5 percentage points less likely to publish. The winners actually do abandon their projects sometimes as well. So if you're the first team to release, about 12% of the time they don't publish, but the scoop teams are a little more likely to abandon their projects. This is not a large effect. It's statistically significant. You can read this as a standard error. But in terms of magnitudes, it's somewhat limited, it's somewhat small. In part, we think because by the time you've deposited your project, you're probably close to publication. And even if the top journals aren't taking it because you've been scooped, there's probably an outlet that you can find for it. Indeed, if you look at what we've called a top 10 journal, which is basically Science, Nature, Cell, and a few other top field journals, the winners go into those journals about 32% of the time. And the scoop papers are about 6 percentage points less than that, or about 26% of the time in one of those top journals. So again, there is a penalty in terms of journal placement. In some of the rest of the paper, we actually show a little more detail about how this happens, but it seems like editors would prefer not to publish scoop papers. But of course, sometimes these races are very close, and so the second team is able to get their paper into a good journal within a few weeks or months. And then lastly, if we look at citations, we do a log transformation. So the winner gets about 29 citations on average, and you can interpret this as a 20% lower number of citations for the scoop projects. Another way to interpret that, if we just plot the levels, this is a simple bar graph where the winner is getting 29 citations and the loser 20% less than that is about 24 citations. So looking at the plot, of course, this looks like it's much more evenly distributed than we might have thought. And indeed, the winner gets about 54% of the citations. Now, when Carolyn and I first found this result, we were sort of interested in benchmarking this to people's expectations, because obviously, if the threat of getting scooped affects behavior, it's really important to understand what people perceive to be the threat. And so we did a survey. We sent out an email, actually, maybe some of you were in this survey, an email survey to the corresponding authors on papers linked to the PDB since 2014, about 9,000 emails. And what we did is we posed a hypothetical scenario. So suppose you've just completed a very promising research project and you plan to submit it for publication this week. What's your probability of getting scooped before that publication comes out? So we're trying to frame the respondents to think of these late stage cases that's similar to what we have in the PDB, and then ask them what the probability of getting scooped is. The follow-up questions then were, suppose that you did indeed get scooped and that the winning paper came out in Science and got 100 citations. So then the question is, what's the probability that you would still publish? What's your expected journal placement, and how many citations do you think you would get relative to the 100 citations of the winner? When we asked that, they were much more pessimistic than what we find in the data. They reported about 43 citations as their expectation, and that would correspond to a winner share of about 70% of the total citations, which is much higher than the PDB estimate. Respondents were also very pessimistic about their probability of getting scooped. They thought it was about 2 or 3 times higher than what we actually find. Also about the probability of publishing altogether. And they were mostly pessimistic about the impact factor of the journal they would place in. So if we said the winning paper came out in Science, they would often say, well, I'll probably end up in +1 or other lower ranked journals. In reality, we see a lot of these scoop papers because they're about important topics. They can end up in journals like PNAS or JMB or other outlets. So it does seem like editors are willing to publish some scoop papers. And so the impact factor effect was not as large as the scientists expected. So just briefly on the long run consequences, we wanted to look at whether getting scooped affects your activity in publishing and in the PubMed records and PDB 5 years later. We look at the total publications that you have in the next 5 years, as well as the total citations to those papers. And that excludes kind of the winning or the scoop paper. We're now looking just at subsequent work. So we wanted to do this separately for novices and veterans because, a lot of the concerns in science about early career setbacks suggest that novices are at much more risk of maybe getting pushed out of the profession if something bad happens. And so I think you'll find most of the angst about getting scooped is coming from young scientists, which is understandable. So we'll look at the effects separately for those two groups. So first, looking at just all scientists, if we look at whether they're actively publishing in papers in the PubMed record 5 years later, we actually find no significant effect. It's negative, but when you look at the standard error, it's not statistically significant. We can't reject that the effect is 0, basically. On the other hand, they are less likely to be active in the PDB 5 years later. So it's possible that even if you're not discouraged enough to drop out of science altogether, you might change the direction of your research. Maybe instead of doing structural biology, you're doing sort of a related set of experiments, and so you're less likely to end up in the PDB. But in terms of the publications, we still see about the same number of publications statistically, and even top publications. We do find significant effects. We use, as I should explain, we use these 3 stars again to suggest that it's statistically significant. There is an effect on citations to those future publications by about 17% fewer. If we split this up by novices and veterans, you'll see that the effects are mostly driven by novices. They're a little more likely to change their course, move away from the PDB compared to the veterans. And the citation effect is really driven mostly by novices. So the veterans have only a 13% penalty to their future work, whereas the novices have a higher effect. So that, again, I mean, that's cause for concern for some people. You feel like if you get scooped, you may be getting less attention, not just to that paper, but to your future work as well. Or maybe you feel like you're behind, maybe they're more likely to change course, and those types of things seem to spill over into their future citations. So related to that idea is how does science have inequality of attention? There's a lot of complaints about, whether high-profile scientists get cited for the quality of their current work or for their reputation rather than the quality of their current work. If there's a famous scientist at Harvard, we tend to just pay attention to their work because of their reputation. We can really look at a very unique case here where we see different races between high and low reputation teams. So we can find cases where 2 teams are working on exactly the same protein and producing the same results, but one of them might get cited more or less. And so we can use these races to study that. So I'm going to do something very similar here. To define reputation, I'll just go back to our predicted citations earlier. And I'm going to split up this set of races into 4 quadrants. First, we have these high versus high matchups. So this is like a Harvard team racing against an MIT team. We have a lot of cases where low-ranked teams end up racing each other. The interesting cases for us are these off-axis ones, where here the Harvard team scoops the lower-ranked team, but here the converse happens. So here the Harvard team is getting scooped. And what I'm just going to do is take those 4 quadrants and show the difference in actual citations in those races. So first, if you look at the evenly matched races, when a high team scoops a high team, they get the winner gets 55% of the citations, and that's the same with the low versus low. And so that's very similar to what we saw on average. Of course, the level of citations is different here, either because of their reputations or because they're working on different types of projects. But the distribution within the race is what we care about, and that's pretty constant with what we've seen before. Now these mismatched races, though, are more interesting. So here the high-ranked Harvard team scoops this other lower-ranked team, and on average they're getting 66% of the citations, much higher percent than we saw on average. And the converse, maybe slightly depressing for some of us, but maybe not surprising, is that even when the low-ranked team wins the race, they get fewer citations than the winner, so about 46% on average. So even when you scoop a Harvard team, they'll probably end up still getting more citations than you. So we think that this is really interesting. It's important to understand, in terms of policy about journals and funding of how different scientists have different access and reputation. And so we think that these results are providing some extra evidence of that. Okay, so I'm going to hand it over to Carolyn. We have about 5 or 10 more minutes before the Q&A. So we're just going to preview this next project, which is called Race to the Bottom, Competition and Quality.
Yeah, so just to give everyone a little bit of background, I think just Ryan and I are just very interested in how competition manifests itself in science and what are the implications of science being such a competitive field, and sort of broadly, do we think this is good for science or bad? And I think that's a very hard question to answer, but we're trying to sort of break it down into smaller questions and chip away at it a little bit. So that's just sort of the angle we're coming from. And this is sort of more preliminary work. And so just very curious for your feedback, since you all are much more experts in the field than us. So just definitely make a note to tell us if anything doesn't resonate with you or you kind of see any problems here. But broadly, I think, you know, we think of competition in science as potentially being a double-edged sword. So on the one hand, when scientists compete to publish their findings first and establish priority, this can A, increase the pace of innovation, and B, it encourages scientists to disclose their work promptly, because it's only by disclosing work that they're going to get the credit. But on the other hand, there may be a dark side to this competition. And one potential dark side that, you know, we've read a lot about, and I think makes a lot of sense, is that if being first is so important, then scientists may cut corners and actually reduce the quality of their work in their pursuit to publish first. And so actually there's just to give you an example from another field, Svante Pavo is an evolutionary geneticist, and in his book, he has a very nice quote about racing to publish the genome of the Neanderthal and having this competitor, Hendrik. And he sort of just very succinctly says here, Hendrik's paper illustrated a dilemma in science. Doing all the analyses and experiments necessary to tell the complete story leaves you vulnerable to being beaten to the press. Even when you publish a better paper, you are seen as mopping up the details after someone who made the real breakthrough. So this perfectly illustrates, I think, the tension that we hope to explore, which is this idea that as a researcher, you want to do a good job, but you know that if doing a good job is time consuming, you're opening yourself up to the risk of getting scooped. And so then you sort of feel this tension between quality and, you know, wanting to get priority, and that may reduce the quality of the work you end up publishing. So just to very briefly tell you a bit about how we're trying to approach this problem in structural biology. What we do is develop an economic model of competition and racing in science. And the key insight from this model we develop is that some projects are high potential and some projects are low potential. And what we mean by that is that before you start a project, you have some notion of whether, if you succeed, this will be a really high profile study that, you know, ends up in Science and will get a lot of citations. You know, maybe you're working on, you know, a protein that's very relevant in disease. And then on the other hand, some of your projects you might know are not going to garner as much attention. And so we call that a low potential project. And the key insight from our model is that if scientists are concerned with maximizing their citations and reputation, you're going to get more people trying to work on these high potential projects. So in turn, these high potential projects are going to be the ones where the risk of getting scooped is the highest. So these are precisely the projects where you should expect to see the most rushing and then the largest negative effects on the project's ultimate quality. And I think this is like not a terribly unintuitive sort of set of predictions, but they're very hard to test in most fields because there's no notion of being able to measure the quality of execution of a project. You know, you could look at citations, but in a field like economics, that doesn't tell you very much about how carefully a project was done. It just tells you that, you know, this was a topic that lots of people were interested in. The PDB is very special because in the field of structural biology more generally, because there have been developments of these measures of project quality. So that's really why we are excited to focus on this field. So just to sort of tell you how we construct from the PDB some of the things we need to test these ideas. The first is this measure of project potential. And what we're able to do here is take the very rich data of the PDB, features like the protein's classification, the gene it's linked to, et cetera, and actually use those to predict how many citations we think the paper's gonna get. And papers that are predicted to get a lot of citations, we call these high potential proteins. We are actually able to see a little bit how competitive the structures are using, again, these BLAST clusters and seeing whether the same structure or a highly similar structure was repeatedly deposited in close succession. And that's a clue to us that yes, a lot of people were working on this structure. We can get some insight into how long people actually work on these structures by looking at the time lag between the collection date and the deposition date. And then lastly, we actually have these quality measures because there's, you know, these things like refinement, resolution, R-free, and these different outlier measures. And so what we actually are just going to do in this talk is combine these three measures here, and we'll standardize everything just so that higher numbers actually correspond to higher quality, even though sort of in the raw data, like for example, lower resolution numbers are actually better. Here we're just going to standardize everything so higher values corresponds to higher quality. And so I know this was fast, but just to sort of show you some of the results we've generated, the first thing we can see is that in fact, yes, projects that we predict to generate more citations, so these high potential projects, are more competitive. We do see more people working on them in close succession. So that's the first result. The second result we see is that these projects also appear to be completed faster. So this is consistent with this idea that people are aware that they're working on something that may have a lot of competitors, and so they actually finish the projects faster. And then lastly, what you see is that these high potential projects are ultimately lower quality. And so I think this is a very interesting result. This is a result we also are cautious at least to interpret, because I think there's a natural concern here that these high potential projects, are they lower quality just because people are rushing, or are they lower quality because they're more difficult in some way, and that's what drives the lower quality? And so I think we take this very seriously. I think the first thing we could point to is, well, they were completed faster. So that does tell you something about rushing. It does seem like it might have been at play. But then the second slide is that one interesting thing you can do is compare how these scientists behave who are working in university settings to scientists who are working at structural genomics consortia. So these scientists are different in several ways, but I think one interesting way in which they differ is that the scientists who work at these structural genomics groups are less focused on publication. And you can actually just see this very cleanly in the PDB data, because about only 20% of the structures solved by these groups have an associated publication. So if they're less focused on publication, then racing for priority seems like it could conceivably be less of an issue. And so if you just plot the relationship between quality and potential separately for these two groups, you do see that it is much less downward sloping for these structural genomics researchers. And so to us, that is indicative that at least a significant portion of this negative relationship is likely driven by competitive concerns. And so we sort of see this, then we sort of, our takeaway overall is that yes, this sort of proposed dark side of competition, that it reduces the quality of scientific work, does seem to be a legitimate concern. And so, you know, is something that I think we should take into account when we think about how do we want to set up incentives in basic science. And I think it's very relevant also for these sort of newer forms of scientific research and collaboration that we're starting to see in the wake of the Human Genome Project, ways of making science more collaborative and less competitive. And, you know, that actually there may be some serious benefits from that. So I'll turn it back to Ryan.
Good. Yeah, thanks. So I mean, just to summarize, you know, the first project, we do find that there are effects of getting scooped. I mean, so there's reason to be concerned, although we think our results suggest that probably people are more competitive than they need to be. They're sort of worried that the costs of getting scooped are higher than we can actually find in the data. And then in terms of normative implications or really policy implications, you know, we could think about whether this premium for priority is too large or too small. Again, it could be good because it incentivizes effort. It encourages people to put their papers out. On the other hand, they may be racing at the expense of quality, and those are things we might be concerned with. There are a lot of journals, by the way, who are sort of thinking about this. Some journals like eLife and PLOS Biology are putting in sort of scoop protection policies where if you can, you know, get your paper in within 6 months of getting scooped, they'll still publish it. So we know that editors and other policymakers are thinking about some of these features, and we hope that the data that we've looked at from the PDB and the conclusions we find are kind of helpful in furthering that policy discussion. So thank you very much for listening, and we're happy to take some questions.
Thank you, Ryan, and thank you, Carolyn. That was great. That was really very interesting. So I'm going to hold my questions to our meeting with the graduate students at 1:15, and the floor is open to anybody who'd like to unmute their mic and ask a question.
David Haas here. I have a question. Have you ever seen a tendency where people fail to cite either a previous publication or a superior publication and have the paper published simply because it implies priority?
That's a good question. So, I mean, part of this is about whether editors are good at kind of assessing priority. And I don't know, some scientists might try to sneak their work in. It sounds like, do you mean citing their own previous work or maybe a competitor's work?
A competitor.
Yeah, I mean, I'm sure that does happen. It's an imperfect system. One thing we always come back to when we're discussing this is, unlike a patent system where there's a legal framework for who should get credit, maybe a judge or an examiner is making that decision. In science, it's more of a norm, a community, norm of assigning credit based on publications and citations. And so it's it can sometimes be more of a Wild West. We know people complain about things like that. We haven't looked systematically in the data to try to find those effects, but it's something we could look at.
Okay.
Thank you.
I have another question if no one else. Oh, go ahead.
Oh, go ahead.
This is Andy Bird. I'm at the NCI, not at Rutgers, but I've been associated with the PDB for a long time. And so I thought this was a fantastic view, and digging into the data and using this, both is interesting to me as a structural biologist, but it's also really a fascinating sort of ongoing impact of the PDB, where it shows where, because of the quality of the data and the vastness of the data, you can go back and you can mine this and do this. So it's very interesting. And I think I passed my comments to Stephen already that I think this would be great information for tenure track investigators coming along to look at this and to have a discussion about what they're seeing and how they're kind of freaking out about how this is going. And so I hope all the graduate students take this to heart. And I think, so I guess in general, I'm wondering how you, so you use the data up until 2017. And so you have a little bit of predictive, but is there a way that this can be something that you revisit in segments of time so you can get a trend line of how these mores and behaviors are changing? Because I think I would say, having, you see my gray hair, I've been in this for a while, and I would say that pressures have changed through the course of our career and with the technology. As you mentioned that, there are new techniques now and you were able to focus mainly on crystallography data, but I think this evaluation of the data could be very interesting across pieces in time as you go through this. I wonder if that's possible to do with your model.
I think it's interesting because it's surely true that the probability you get scooped has gone up with time. Just if you look at the rate of, there's so many more people depositing, per year now. So that side of the concern has certainly gone up. How bad it is conditional on getting scooped, I don't know. And we could look at heterogeneity along those lines too. But I think that's very interesting, because when you read the piece, like if you read sort of the literature, people talk about how the competition is getting, more and more fierce. And so I think being able to say something empirically about that would be really interesting.
I think as an auxiliary to that, I don't know how hard this next point would be, but is there a way to correlate that with funding through the publicly accessible data at the NIH, for example? That would be perhaps terrifying to tenure track investigators, but also perhaps you may find the probabilities are more like what you saw in the real data as opposed to the perceptions. And that could, somebody probably buy you drinks for that.
Yeah, that's fun.
The pay lines at the different NIH institutes, yes. Yeah, we've looked at that a bit.
Yeah. Chenghua Xiao, I think you had a question?
Yeah, so I have a kind of a comment or suggestion and a question. So the comments actually, I want to present my case that was the second research project when I was in graduate school was actually scooped. And I actually, when I was listening to you, I did some research on that project. And I realized that something maybe by demonstrating this case, I can show you some variables you can consider in your further investigation. So before I joined PDB, I was studying in Boston University, and the second project was on lactobacillus protein in milk. And it's at the same time, a Harvard group was doing the same research on the structure. And actually we saw the structure at the same time because we presented the structure at the same meeting in one session. I mean, before and after. So, but we published much later because the first journal we submitted just turned it down. And the structure actually is 3BN6, which is ours, and 2PQS, which is Howard. But I look at the citation as of now, ours got much higher citation, even though we come from a smaller group and we published later. And the journals published both structures were about the same tire. Where ours got 97 up to today, and the Howard group got only 49. And the reason is, number one, our structure was in a much higher resolution. Higher resolution means we provided more details from molecular level, from atomic level. And second is because after we were turned down for the first journal, we did some biochemical studies to study how this protein binds to membrane. The protein can bind to membrane in a reverse way, so that it has more contents in the paper more than the paper published by Howard. So my question is that other than the sequence blast, did you consider any other variables? For example, whether or not there's some ligands, or whether or not the publication was exclusively on the structure, or is just part of the structure? Those may actually be the reason that you don't find very significant differences for the citations.
Yeah, no, those are all really insightful comments, and I mean we've sort of thought through a lot of those same types of variables across these papers. So I'll say a few things. One is that we are finding cases where there's a lot of overlap between the papers, but not perfect overlap. So that, if we lean on the sequence similarity just to make sure that they're sort of talking about the same protein. But you're right that some of these papers have different, different ideas or sort of experiments that they're reporting that could be different. So one thing is that we think that on average, at least at the very end of the race, those things are sort of evenly distributed between the teams that win and lose. And so that's not as much of a concern for us. But once that first paper comes out, as I was saying before, the second team could decide to make changes to their paper. They could decide to either improve the quality, they could add some biochemistry, they could do things like that would mitigate the cost that we're looking at. So we can't, we're not shutting that down. We're sort of just finding what's the overall effect of getting scooped. It could be much higher if you just tried to just publish the paper as is, but we do think that there's probably some of that going on in the background. And we haven't shown much of that in our results, but we could look, for example, at whether the higher quality structures might, sort of ameliorate any cost of getting scooped. We think that that probably is true. On average, it looks like the winning and losing papers have the same quality on average. Some, it's always one is better than the other, but so I don't know if the quality dimension is a main channel, but if we did more like text analysis, for example, we might be able to learn a little bit about what you're talking about.
Yeah, for iterative structure, usually the resolution is one factor that impacted citation. So yeah. I'm just something like.
So Chenhua, I think we're going to have. I'm sorry, I think we're going to cut you off. We're going to have to bring this to a close. It's 1 p.m. and I want to be respectful of everybody's time and the fact that many of us have to jump on another Zoom meeting, if not at 1 p.m. very shortly thereafter. So on behalf of the RCSP Protein Data Bank and the Institute for Quantitative Biomedicine, I want to thank you, Ryan, and you, Carolyn, for a great presentation on a very stimulating topic. Absolutely. That, as Andy Bird indicated, would be of high interest to the structural biology and more generally biomedical research communities. And I see David Haas is giving a thumbs up, and I'm going to do the same. That if you've got reactions, you can provide your encouragement for our speakers. And
Thank you very much.
We will follow your careers with great interest, and we hope that you both continue mining the PDB either on a collaborative basis or in independent projects. And finally, my thanks to everybody for participating today. The number of participants exceeded 50. And I hope we'll continue to show an upward trend in participation now that people are getting the hang of this online IQB Wednesday seminar series.